Hello, biology. Welcome. I'm sorry I can't be with you today, but the good news is a lot of this will be review for you. So hopefully this will help with all of that. So the first thing we're going to go over is hypotheses, theories, and laws. And you probably have heard of all of these things, but there can be some misconceptions, so I'll make sure to clarify those for you. So a hypothesis is an educated guess based on your previous knowledge. Maybe it's from observations that you've made, maybe it's from looking at previous research, but based on what you know, the hypothesis is an educated guess. Meanwhile, the idea of a law in science is an observation about the world that occurs so often we accept it as a fact of nature. So an example of this might be the law of gravity. We know gravity pulls us down towards the earth. The force of gravity is a downward force, and we know that anything that goes up is going to get pulled back down by the force of gravity. And so that's what we consider to be a scientific law. And then finally, we have what's known as a scientific theory, and this is an explanation about why a law occurs. The idea of a theory is that it's been tested so many times over and over again that we accept it as the truth. Now, any of these things, we might discover something else in science later down the line that changes what we think with a hypothesis, with a law, or with a theory. But all of them for now are based on what we currently know. So, for example, the theory of evolution is an explanation for how species have changed over time. And it's been tested over and over and over again. And that is what we have come to find about the change of species over time so far. That's not to say we won't find new information, but the theory of evolution, it's not like a conjecture, like how you might think about theories in other disciplines. A theory in science is an explanation for something that happens in the world that's been tested many, many times. So laws are observations, theories are explanations. Graphs are used to show the overall picture of your data, and they're kind of like a way for us to display data visually. And there's a lot of different types of graphs. Right over here, this is a graph showing the levels of carbon dioxide at Mauna Loa, one of the volcanoes in Hawaii, from 1960 until the early 2000s. This would be like a line graph, and line graphs are often used to show change over time. That's why I like to say time, line, to help you remember. Over here we have a bar graph, and this is showing the population of wolves, the number of wolves in the different populations in and around Yellowstone Park. So bar graphs are a great graph to use when you're comparing between different groups. So you can see we have 3 different groups here: the wolves in Yellowstone National Park, the wolves in the northern range, and the wolves in the interior. And we're comparing the populations of those different wolves. So that's why a bar graph makes sense, because we're looking at different groups. So like we mentioned, the line graph shows changes over time. Like I said, line, time, they kind of rhyme, so you can remember it that way. This one right here is an example of a scatter plot. And scatter plots usually show the relationship between the independent variable on the x-axis and the dependent variable on the y-axis. And it might not be a perfectly linear relationship, which is why we can't necessarily connect it with a line, but we'll just plot the points and see if we notice any trends. Like in this one, it's age versus size. We don't know much else about it, but we can see as the age gets bigger, the size gets bigger. It's not a perfect line, but it's trending. Like we mentioned before, bar graph, one of the other common types of graphs, is used to compare different items or groups. So maybe if I wanted to compare, say, what is everyone in the class's favorite social media, favorite form of social media, those would be our different groups, and then we could compare the numbers of people that liked those different things. And then the last one that we'll see a lot in biology is a pie chart. And pie charts show proportions relative to a whole. So out of 100%, you can kind of figure out how much of the pie is taken up by each of the different test components. So you can see like here, the blue group probably takes up over 50%, maybe even close to 60%. And then the little tiny pink group over here is the smallest percentage. When it comes to graphing, there's a couple key things that you need to include. So I like to say you should make your graph talk, T-A-L-K. So T stands for title. You always want to give your graph a title about what it's telling the audience what it's about. A stands for axes. So on the axes, you need to make sure that the x-axis is used for your independent variable, and the y-axis that goes up and down is used for your dependent variable. And a good way you can remember that is by using the mnemonic dry mix. So dry stands for the dependent variable, which is the responding variable or the result, goes on the y-axis. And the independent variable, which is the manipulated or variable that's changed, is plotted on the x-axis, hence the mix. So manipulated, independent, x, dependent, responding or result, y. So here's the x, here's the y. So you would put your independent variable here, your dependent variable here. Another big thing that you should make sure is that when you're graphing, the values take up the majority of the graph. If your graph is really tiny and you make the values of your numbers really, really small and the whole graph is taking place in this one little corner, it's a lot more difficult to see the changes and the trends when you're looking at it small. If you can blow it up to take up the whole page or as much of the page as possible, you're going to be able to see the graph a lot better, see the changes, and not make as many mistakes. If you can make the values better by not starting at 0, you can create a little break in the axis by creating a little slanted double line and then start wherever you want to start the graph. So let's say I wanted to start at like 50. I could do 2 little slants here and then start this at 50. And then when we talk about talks, we did title, we did axes. Now we're going to talk about labels. So you want to make sure that you label each axis with what it's showing. And if you have the measurements, you can show the measurements there. So for example, if I wanted to see how many inches my plant grew, I might call this one plant growth, and then in parentheses write inches. And then down here, maybe I would say like amount of sunlight in amps or something like that, however I was measuring it. The K stands for key. You don't always need a key, but sometimes it's helpful to include a key in your graph. It depends on the situation. So you don't always need a key, but if it would help your reader, you can make one, telling them what the colors stand for or what the different lines stand for. And then the S stands for scale. Your graph needs to be to scale, and that's kind of what we're talking about here. So each interval as you go up the graph needs to be consistent. It needs to go up by the same amount each time. So that's for each particular axis. So let's say I'm on the y-axis. Maybe I could go by 5s, like 5, 10, 15, 20. Maybe I could go by 2s, 2, 4, 6, 8, 10. But whatever you choose for your scale, you want to make sure that your graph is taking up as much of the space as possible. So T stands for title. You want to give your graph a title. A stands for axes. You want to make sure the independent variable goes on the x-axis. The dependent variable goes on the y-axis. L stands for labels. You want to make sure you tell us what we're looking at on either axis. K stands for key if you need one. And S stands for scale, meaning the intervals between the numbers on both axes need to be separated by the same amount. And you also need to make sure that your graph takes up as much space as possible on the page when you're making your scale. So that's a little bit about graphing. Oh, there's our title. There's our label. There's our dependent label. And in the next section of the video, I'm going to explain a little bit about how to write a lab conclusion. Okay, for the second part of this video, we're actually going to talk about how to write in science. And we are going to use a specific method in this class known as the CER method. And so that's why this is called Writing in Science? Certainly. Okay, CER. So CER actually stands for claim, evidence, and reasoning. And so I want you to take a moment right now and think about, what is a claim? If I were to ask you in class, what is a claim? What would you say? Think about that. What about evidence? What would you say could be considered evidence? And then finally, what would you consider to be reasoning? If you need to pause and think about these, you can. Officially, we're going to state that a claim is a statement about the results of an explanation, or about the results of an investigation. So let's say you do a lab experiment. There's always going to be a question that you're trying to answer in the experiment, and the claim is really the answer to that question. Your evidence is going to be the scientific data that you use to support the claim. And that evidence should be sufficient, so it should be enough to make you feel like the claim is supported. It should be appropriate, so it should be relevant to what you're talking about and support your claim. And you should have both qualitative, which is your observations, and quantitative, using data or numbers. Both types of those evidences in your explanation if you can. And then finally, we have reasoning. And reasoning is really like what I like to call the so what of CER. It's where you basically explain how your evidence helps support the claim using the information that we've learned in class. So you can use the scientific principles that we learned in class to explain why you think you saw what you saw. And that's the reasoning. Here are some common misconceptions about CER. So one is that the hypothesis is not the claim. A lot of people think that, but the hypothesis is done before you answer the question or before you even do the experiment. Sometimes if your hypothesis was correct, you can use some of the wording from your hypothesis to write your claim, but that's not always going to be the case. So the hypothesis and the claim are two different things. And we'll talk about the hypothesis a little bit later. The claim is an answer to the question you were trying to answer in your experiment. So a good way to test if you've written a good claim is to ask yourself the experimental question and then read just your claim and see if that's a satisfying answer. For example, if I were to ask you, why is the sky blue? And your claim was, the sky is very pretty today. Do you see how that doesn't really answer the question, why is the sky blue? So that's a good way to test your claim. Your evidence can be scientific information. So for example, it could be research or prior knowledge that you have. It can be information that you've learned in this class, or it could be experimental data. So you could use data from your experiments to support your claim. And anytime you have the opportunity to use quantitative evidence, make sure that you do. That is the strongest piece of evidence that you can use, because then you have actual experiments that can back up what you're saying. Like we mentioned before, the reasoning is really the explanation of how the evidence supports the claim. You're telling us why that evidence means that you are correct. And so what you want to do in the reasoning is make sure you use and rely on the science that you've learned. Some of these early experiments, you might not know a lot of science yet, and that's okay. But as we go throughout the year, we want to be able to tell that you took biology freshman year in your answers, because you're able to explain why the evidence supports the claim using the scientific principles. And if you do this well, your CER can actually be a really good conclusion to a lab experiment. So next class period, we're going to do a lab, and we're going to get to write up what we've learned. And this is going to be a great way and great model for you to do that. So we are going to watch a video now about Space Alien Girl. This is one of my favorite videos. And she's making an argument. And her argument is going to answer the question, is your dad a space alien? Even though she's only 6, we are going to grade her like she is in high school biology class. So as you're watching, try to identify her claim, evidence, and reasoning. And I do recommend watching this video twice through, because the first time you might be distracted by how cute she is. So make sure you watch it twice through to make sure you really understand what she's saying. At this time, if you're following along on here, that's totally fine, but you're going to want to pull up the Google Doc version of the slides, which is the first link of the slides on our class website, so that you can watch the video. So you can pause this here. I'll give you a second to watch the video, and then we'll talk about what's next. This is what the video looks like in the slides, so hopefully you watched it. All right. Okay, so now what you need, what you're going to want to do is analyze her argument. So Mr. Romero handed out slips of paper that include her entire argument. And so on your copy of her argument, I want you to highlight her claim. What was she claiming? What did she decide about her dad? That's the first step. Next, I want you to underline her evidence. So what information or data did she use to support her claim? Finally, I want you to put a star next to her reasoning. How did she explain her evidence, if any? You can pause here if you need more time, but we're going to move on. So now the other piece of paper Mr. Romero's handed out is a rubric, and this is a very similar rubric to the one that you'll be graded on for many of your labs. I would like you to take a moment now to read through the rubric and see what's expected for each number of points. So for example, in order to get a 4, she has to have done all of these things. She has to have written a claim that answers the question. She needs to write it as a complete and complex sentence, and no explanation is given with the claim. So she doesn't say because in her claim. She saves the because for her evidence. That would get her a 4. So you can read each of the criteria and give her a score. And then I want you to, on the rubric, you can write it directly on the thing. I want you to circle or write what her score is in each category. What would you give her claim? What would you give her evidence? And what would you give her reasoning? And make sure that you can explain why. So you can pause here and take a second to do that. And then think about, what does she need to do in order to get 4s on everything? We'll talk more about that next class period when I'm with you, but the last couple things I want to teach you about are your hypotheses. So our hypotheses in high school are getting a little bit more advanced than the typical if-then method. Always make sure that you make your prediction for your hypothesis before you start the experiment. And then after you make your prediction, include an explanation for why you think what you do. So make sure that you both include what you think and why you think that for your hypothesis. And the last thing we're going to ask you to analyze in your experiments most of the time is your sources of error. It's important to note that no experiment is perfect. There will always be errors. Some of these things are things that we can't control, like maybe there was a fluke power outage and your something that you needed ran out of batteries for the experiment or ran out of power. Sometimes they can be errors in the experiment, like maybe you made an error in your measurement or you made an error counting something. And then there's human errors, which are errors related to the human performing the experiment. So maybe you had a fluke day where we're going to do a typing lab. Maybe you had a fluke day where you were really distracted and your typing was way slower than it normally is because something was on your mind. That could be an example of human error. So some of these errors are things that we can take precautions for so that when we repeat the experiment in the future, we don't get the same influence on our data. And so there is always a lot that can be improved for experiments, especially ones that we do only once or twice in the classroom. But one of the best ways to correct for errors if you're trying to improve the experiment is to repeat it multiple times for many trials. And that way you can find out if the data that you found the first time is the same the next 3 times you do it. Another way you can get multiple trials, it's not the most scientifically perfect way, but another way you can get multiple trials is by averaging the data from you and multiple other people that have done the experiment. And that can also sometimes help you avoid the error. So that's some information about error for when you get asked about it. That's pretty much the end of the slideshow. We will be going back to our space alien girl and reviewing how we graded her next class period. But if for today, if you can please take a look at your homework, it will be Reading Check 1.2, SPS 1.2, and 1.3, which is about graphing, and then KEO 1.2. And that is your homework for the rest of the day. So thanks, biology, and I'll see you next time.